Okay, we left off on Friday talking about how it is that bacteria have evolved to take advantage of mutational events to help them survive varying environments. So the example that I used initially that I was rushing through towards the end of lecture was how a bacterial protein such as this illustration of SAB-A could vary the length of this repeat, this dinucleotide CT repeat, to turn the protein from either being on or off. So phase on or phase off. So this is phase variation that actually functions kind of like an on or off switch. Any one bacterial cell will either express the full length SAB-A or will not. There is no intermediate between those two, phase on or phase off. And that's being mediated by slip strand mispairing events that change the length of this CT tract length. Now, this particular gene is a really good illustration, and why I use it is because it has yet another region associated with this gene that also is involved in phase variation. But it's not an on-off switch like the poly-CT tract, where the protein goes either on or off. This is more like a rheostat or a dimming knob. There is a polythymine tract that's now in the promoter region of this same gene, this SAB-A gene. It's upstream of the coding region, so this pink area is where that poly-CT tract was, that fuchsia box. Now we're talking about an area of about 18 or so tandem thymidines right in a row. Here's the minus 35 box of the promoter for this gene. This sequence, because it's repetitive, but now the repeat length is 1, this will also vary in length during replication. Any individual cell within a population has a fixed number of thymines, perhaps 18. But at some frequency, when it spawns a daughter cell, that daughter cell may have 19, or it may have 17. So there's going to be variations in this poly-T tract, which are going to affect the initiation of transcription of SAB-A. So the previous example that we used was an example of translational phase variation. The phase variation depended on the phase of the coding region. Here, this is transcriptional phase variation, and it just so happens to be controlling the same gene. So that based on the length of this poly-T tract, regions upstream, which could affect transcription by binding transcription factors, are now moved further away or perhaps pulled more closer to the RNA polymerase binding site, the promoter site. And so either turn up the transcription slightly or turn down the transcription slightly. So it's not an all or nothing, it's a volume switch. And so the combination in this particular gene of the ability to turn the gene on or off completely, as well as to change the volume, gives you an idea of how important the proper expression level of this gene is, because phase variation controls two different aspects of the expression of this particular gene. These are both examples of phase variation. But now I want to talk about a particularly fascinating aspect of phase variation, a minor variant, if you will, of phase variation called antigenic variation. In the phase variation that we were just talking about, we're talking about one protein. That protein either being expressed or not, or perhaps when it is being expressed, perhaps expressed at a slightly higher level or a slightly lower level. But it's always in dealing with one protein. When we're talking about antigenic variation, we're talking about the variation in terms of at least two proteins. One being expressed at one time and another protein expressed at another time, but neither protein are expressed simultaneously. So some bacteria have the ability to switch back and forth between two different genes randomly, and then the environment allows them or selects for whichever particular population is best fit. So antigenic variation is the best known example of this, or what I think is the best studied example of this, is actually in a bacterium that you're all familiar with. Hopefully none of you have been infected with, Salmonella enterica. So that famous cause of food poisoning and gastroenteritis. Salmonella enterica actually has genes that encode two different forms of the subunit of its flagellum. The subunit is called flagellin, and it has two different genes, and it either expresses one or it expresses two, but it never expresses one and two at the same time. It has the ability to switch back and forth between those. That switching occurs randomly in a population of Salmonella, either in the laboratory or during infection. Which population predominates depends on the environment. So if you as a host create an antibody response, an immune response against one form of Salmonella's flagella in the course of trying to clear the infection, you can, in theory, kill off most of the Salmonella that bear that flagella. It's a little too much rhyming there. But if some small subunit or some subpopulation is expressing a different form of the flagella, that antibody can't touch that population. So that population can survive. So the bacteria may be able to persist in your gastrointestinal tract for slightly longer by having two different populations. So until you've formed an antibody response against both forms of the flagellin, perhaps the bacterium is capable of holding on for another few days, which would, from the bacteria's point of view, potentially infect a few other people in your dormitory or in whatever restaurant you happen to be working in, or things like that. But as I said, only one type of that flagellin protein is expressed at one time, never both. And the variation is a random event. There is always this idea that people have, a misconception about how evolution works, in that if I tell people that if you make an antibody against one form of the flagellin, people will think, well, that forces the bacterium to switch to produce the other form of the flagellin. And that's not true. The switch is completely stochastic. It is completely random. The bacterial population will, at some frequency, just without any prompting, without any design, will produce some fragment of the population which is expressing the other form of flagellin. And then natural selection occurs. So if you have an antibody against one form, it kills that population and allows the other population to now predominate. But that switch is not driven by any environmental stimulus. It's a very common misconception. This is mechanistically how Salmonella enterica switches between these two forms of flagella. So what's shown here on the top is just arbitrarily, the cartoonist here has chosen to show a situation where the bacterium is expressing the flagellin H2 first. So this gene, again, there's a little bit of too many letters here. They should have just called this gene FLGB. They should have just called it the H2 gene, because this orange box here in A, FLGB, encodes the flagellin, the second form of flagellin, called H2. H, we kind of touched on this a little bit earlier. So H is actually from a German term for flagellin. So you've all heard of the E. coli 0157H7. H is a German term for the flagellar antigen. And so this flagellin is called H2. So in this particular case, shown in A on the top, the Salmonella is producing flagellin 2, H2. Here's the promoter, shown as a P, and anytime you see a bent arrow like that, that's usually a universal designator for the start of transcription. So transcription is beginning here at this promoter, and this wavy blue line is meant to represent messenger RNA. So this promoter is driving transcription of this H2 protein, but it's also driving the transcription of the gene just downstream and in part of the same operon as this H2 flagellin called FLJA. That gene encodes a protein shown here by this red ball, which is a repressor protein. This is the mechanism whereby Salmonella enterica has evolved to make certain that when it is expressing H2 flagellin, it does not express H1 flagellin. That's the gene shown on the extreme right here, FLIC, F-L-I-C. FLIC encodes the other form of flagellin, the other subunit that can make up that long helical power-stroking flagellum that rotates in the bacterium. But in this particular case, when H2 is being produced, the repressor protein, FLGA, is also being produced simultaneously. It binds to a site associated with the promoter for the H1 flagellin, FLIC, and stops its transcription. So very simple to see in A, on the top of this cartoon, how the bacterium expresses H2 and at the same time represses H1. Now, by random chance, at some frequency within the population of that bacterium, there's an event that occurs which is really pretty cool. A segment of the DNA, called appropriately enough an invertible element, actually does exactly what that name suggests. It inverts. The region shown here in A, beginning on the left with this Hicks L gene or sequence, all the way down to Hicks R, that element, that piece of DNA, has the ability to flip within the genome. Without doing any other rearrangements in the genome, it can turn 180 degrees. And it does it under its own power. So within this invertible element, between Hicks left and Hicks right, L and R stand for left and right, kind of that makes it easier to remember, is this gene called HIN. The IN portion of HIN makes it easy to remember. This is the invertase. This is the enzyme that actually mediates the inversion of that segment of DNA. So that protein is expressed under its own promoter here, the promoter for HIN. It produces the protein shown as these green ovals, which have the ability to bind to Hicks right and Hicks left. Hicks right and Hicks left are not genes. They do not encode messenger RNA. They do not encode proteins. They're simply named that because these are the sites that bind to the invertase created by the gene HIN. These proteins will now, bound to Hicks right and Hicks left, actually form a loop in that DNA, break the DNA in two different spots and knit it back together such that the inverted region now goes into the position shown here in D at the bottom of the cartoon. Now you can see that HIN and its promoter are going in the opposite direction. The important point to notice about this is look what happens after the inversion to the promoter shown in A that was driving the expression of the flagellum. The promoter that drives the expression of FLJB and FLJA, the repressor, is actually part of the inverted region. So now, after that inversion occurs, that promoter is now pointing in the 180 degrees in the opposite direction. So in this particular arrangement of that invertible element, the ability to express the H2 flagellum, FLJB, is lost completely. And simultaneously, so is the expression of FLJA. And remember, FLJA encodes the gene that makes the protein FLJA, which represses FLIC. I know it's an alphabet soup. But when you go through this slide a couple of times, it will become suddenly very clear, very obvious. But now, because you cannot produce that repressor protein anymore, the H1 flagellin is now free to be transcribed and translated. So the bacterium can switch almost instantaneously from producing one form of the flagellin to another form. And so now, in any population of the bacterium, there's always going to be, so if you start off with a population that is, you start off with a clonal population. So one cell falls down on an agar plate and grows up into a colony. That one cell had to be in either the arrangement A or the arrangement D. Let's say it starts off in the arrangement A. So the first cell divides into 2, 4, 8, 16. You all know how bacterial asexual division works. At some frequency within that population, the majority of the population is going to maintain this particular arrangement, but some few cells, just by random stochastic means, are going to have reverted to the D, what's shown here in D. So there's always going to be some small population that is expressing the other form of the flagellum. So that if the environment shifts such that one is favored over the other, the entire population is not killed. So it's a very nice adaptation to make sure that the bacterium can utilize mutation, because this is a mutation. You have a heritable change in the DNA sequence. This mutation is actually advantageous. Okay, so we've now looked at a few examples of how it is that bacteria have utilized mutations to adapt themselves to different environments. Clever bacteria. Now let's look at how we, as people, have taken advantage of mutations in bacteria to do our bidding, both in terms of just basic science, but also in terms of service to the human community. So before we get into how it is that we've taken advantage of bacterial mutations to do a service, to do a favor for ourselves, I need to first introduce a couple of terms. And those terms are auxotrophy and prototrophy. So normally a bacterium like E. coli or Salmonella are what are referred to as prototrophs. Prototrophs like Salmonella or E. coli, wild type, can take a simple carbon molecule like sugar, like glucose, and turn it into every one of the amino acids that it needs, every one of the nucleic acids that it needs. It can make everything that it needs out of that single carbon and electron source. When a bacterium can do that, it's known as a prototroph. If, on the other hand, a bacterium has acquired a mutation which makes it incapable of producing one of those amino acids, the only way that bacterium can survive is if you provide it that amino acid in its environment. That bacterium is now referred to as an auxotroph, meaning it has to eat something from outside of its environment. So that's shown here in this cartoon. You can take E. coli cells and treat them with a random chemical mutagen, like nitrosoguanidine, which does DNA damage. And then what you can do is, let's say you're interested, as this potential investigator was who did this cartoon, in trying to examine the nature of lysine biosynthesis. So after treating the bacteria with this mutagen, the bacteria are then plated onto a media that contains lysine. So in case some bacterium in here has acquired a mutation in one of the genes necessary for lysine mutation, it would die unless it was plated onto a media which had lysine. So what's meant here in this cartoon by complete medium means that there is lysine present. So any lysine auxotroph which has arisen is not going to die. Now, what you do is take sterile velvet, the kind of stuff that people used to paint portraits of Elvis on, right? The same velvet. You can sterilize that in an autoclave and make a replica of that plate. So all of the 100 or 200 colonies in a very unique pattern, just a random pattern on that plate, will now be created. You can create a mirror image on that sterile velvet plate. So if you just push it against the plate, not all of the bacteria come off of the plate. You know, enough stay on the plate that you can regrow them in their original arrangement of colonies. But now you can take that plate and push it against new agar plates to create identical images of the original master plate. But in this particular case, what you would do is take that velvet replica and plate it identically on two media that differ in their differ in their presence of lysine. So if you put it onto a complete medium, meaning it has lysine, the exact same number of colonies in the exact same order grow, But at the same time, if you replicate plated them onto a media that doesn't have lysine, you could now get all of the cells that are capable of growing without lysine will still grow. But by comparing the patterns on these two media, you can select for a colony shown here in black, which grows only if lysine is provided exogenously in the media, whereas that same colony didn't grow in the absence of lysine. So this is, in essence, the way that we have, over the years, basically determined all of the biosynthetic pathways for all of the amino acids and all of the nucleotides. People have screened for lysine oxytrophs, for histidine oxytrophs, for proline oxytrophs, and found mutations in various genes that they didn't initially know what those genes were, but they realized later that, okay, if we put a mutation in that gene, we can make a histidine oxytroph. So this is, the piecemeal way that old-time bacterial geneticists and physiologists used to piece together all of these biosynthetic or anabolic pathways to make antibiotics. Very labor-intensive. I'm glad I wasn't even alive for much of that. But somebody, well, not just somebody, a very clever biochemist who's actually still alive today, he's an elderly man, but he's still an active professor at Berkeley, named Bruce Ames, decided that he could use this test to use bacteria to look for the ability of various chemicals to cause mutations. So everyone knows that, with all of the chemistry that we've evolved to do very well, that some of the things that we produce have unintended consequences, some of which cause cancer. And so, we still have to, sadly, do too much animal testing on various chemicals. But Dr. Ames decided that what he could do potentially would be to use bacterial oxytrophic mutants to screen chemicals to see if they had an increased mutagenic capability. So this is what's known still today as the Ames test. And so this has probably saved an awful lot of mice and rabbits' lives, because any new chemical that's proposed to be used as a food additive or an insecticide will go through the Ames test first to see if it's mutagenic. The way that the Ames test works is they take one of Bruce Ames' own mutants. He created a Salmonella enterica strain which could not produce its own histidine. So for the most part, or not for the most part, it did have to have histidine in its media in order for it to grow. And treat those bacteria with potentially mutagenic chemicals to see the effect. So that's what's kind of shown in this cartoon. So Ames's histidine oxytrophs are treated with, or are plated on two types of media. The media on the left has just a small amount of histidine, not enough to allow the bacteria to grow to the point of forming colonies, but just enough histidine to allow them to divide 2 or 3 times. Because in the media on the right, that same amount of histidine is present, but there's also a test chemical in here, a chemical that you might suspect of being a mutagen. The small amount of histidine allows these oxytrophs to divide a couple of times. Because remember, most mutations occur, most chemicals cause damage, and you see the damage as a result of mutations which occur during replication. So the bacteria have to replicate a little bit, hence the small amount of histidine. Now, if you plate both, or if you allow both of those to grow in the incubator, on the left, you'll see that in this particular case, 4 Salmonella colonies grew. So in essence, what these colonies are now histidine prototrophs. They have reacquired the ability to synthesize histidine on their own. So in other words, this is a measure of the spontaneous mutation rate. So these 4 bacterial cells, which resulted in those colonies, just by completely random chance in the billions of bacteria plated on there, acquired a mutation that reverted them back to histidine prototrophy. So this shows the background level of mutation, as opposed to this chemical, or this plate, which had the test chemical in it, where you can see about 30 or so colonies have grown. So if statistics bears things out, what you can tell is that the chemical impregnated in that plate has more of a mutational effect. it has caused an increased mutation rate in the salmonella, and some of those mutations actually reverted the bacterium back to auxotrophy. So this is one way that you can, so this would be a chemical that you would probably not want to put into food additives or cosmetics. You probably don't need to bother to do some animal tests with this because you would know immediately that this is a bad chemical. The test is actually even more complex than this. I got ahead of my bullets. Many of the chemicals that end up being carcinogens that are created and released into the environment by themselves, as we would ingest them, are not carcinogenic. They are not mutagenic. But they become mutagenic as the result of our liver breaking things down. So our liver is wonderful at breaking down xenobiotics, chemicals in the environment to try and detoxify them. But occasionally it breaks down something that's not toxic into something that is toxic. So the Ames test actually has another step to it that's not really outlined in this simple cartoon, in which you would take that same test chemical and treat it with an extract from rat livers, and then take that treated form of the chemical and add it to a media to see if it would cause an increased mutational rate. So it's a very elegant test to initially screen chemicals. It's not screening for carcinogens. This is a common misconception. And it's not screening for carcinogens simply because bacteria don't get cancer. Bacteria, grow uncontrolled as long as there's enough media, and that's kind of already the definition of cancer, or at least one of the simple definitions of cancer. So really what the Ames test is testing for is increased mutagenic activity of these chemicals, not carcinogenic activity. Certain other tests, sadly, would have to be done in animals to see if these chemicals that were positive in the Ames test did in fact cause cancer. So this is an example of how it is that we've used mutations, bacterial mutations, to our own advantage in order to test chemicals. But for the most part, mutations are bad things. I mean, they are the stuff of evolution. But for every good mutation that a bacterium comes up with, or any organism comes up with, probably 1,000 or more occur that are deleterious. So most mutations just need to be repaired if the organism has any hope of surviving. So let's just go through a little bit on DNA repair mechanisms. And so we'll start off with one called the excision repair system, or the UVR-ABC, or probably a better name would even be UVR-ABC-D. There's a fourth protein involved here. This is a very good general repair system. This is a repair system that has the ability to detect distortions in DNA. Picture yourself running your fingers along a string and then suddenly finding a knot that stands out like a sore thumb between your finger and thumb. You know that there's a distortion in that. And that's in essence what this UVR-ABCD system does, is it finds distortions in the DNA, which are generally the result of something, some chemical or some radiation type damage. The example shown in this particular cartoon is a thymine-thymine dimer, that famous UV-induced DNA damage that causes us all kinds of problems. We can't replicate, bacteria can't replicate DNA that has this kind of a lesion. So the UVR-ABC system finds that bump in the DNA, it finds that distortion, it removes a section of the DNA, and then a DNA polymerase 1, actually fills that back in. So using the other strand as a template, so the lesion is repaired, the thymine-thymine dimer is gone. Now, I can't remember who it was, but somebody came up after class on Friday and asked a great question. I don't remember who you are, but remember when we were talking about this slip strand mispairing and the fact that the nascent strand of the DNA can loop out like this and re-base pair with the template to reinitiate. So that would lead to an increased length in one strand. Well, somebody asked, why is it that this loop that's formed, it can't base pair, what happens to it? More often than not, what happens to a loop like this is that it's clipped off by UVR ABCD. So more often than not, when slip strand mispairing occurs, and you're headed towards what would seem like an increase in the length of the nascent strand, that bump in that DNA can be sensed oftentimes by UVR ABC. It can be removed. So if this bump is removed and the new DNA is synthesized, chances are it's not going to make that mistake again, and it's going to replicate the complement of this. And the new strand is going to have 5 T's, exactly complementary to those 5 A's. Now, however, if you look at the situation on the right-hand side, slip strand mispairing in this particular case has thrown the loop in the template strand. So here, the loop is in the original strand, not the newly synthesized mutant strand. So now, this loop would be the target of UVR ABCD. This would be the bump that would get excised, and now the new sequence would be serving as the template. And so this would actually lead to the fixing of a shortened or truncated mutation in that strain. So in this particular case, if slip strand mispairing occurs as it does on the right, UVR ABCD actually helps to fix that mutation. And by fix, I mean make it permanently into the genome, not as in fix, as in repair. But this is more typically probably what happens in most cases of slip strand mispairing. That's why that form of mutation is still quite rare. It's more frequent than a random mutation in a random piece of DNA, but it will occur more frequently than normal. Okay, so sorry for that aside, unless you too were confused about how those systems worked in slip strand mispairing. Let's go back to the UVR ABC system and look a little bit more closely at the mechanism that occurs, because it's not like this complex of 4 proteins is constantly cruising the genome searching for this DNA. For the most part, there's only a small amount under normal circumstances. This DNA repair system is expressed at only very small levels inside a bacterial cell. And the UVR A and B, those 2 proteins, those are the surveillance. Those are the enzymes that think of them as just scanning along the DNA, looking for that knot in the string. When they find it, such as this thymine-thymine dimer, what the UVR-AB is going to do by hydrolyzing with the hydrolysis of ATP, it's going to now recruit UVR-C, and UVR-A is going to be lost. There's a bend in the DNA that's created by UVR-B and C, and that bend occurs at the thymine dimers. Now, the UVR-BC is actually an enzyme complex. It has the ability to break phosphodiester bonds, and it's going to break 2 phosphodiester bonds on one strand. So when you break a phosphodiester bond on just one strand, that's called a nick. So here, about 5 nucleotides downstream of the thymine-thymine dimer, you create a 3' nick. So the enzyme has broken 1 phosphodiester bond. And then it has the ability to translocate upstream of the thymine-thymine dimer and make another nick, about 8 nucleotides upstream. So it breaks another phosphodiester bond on the same strand that the other nick was made, but now upstream, or 5' of that thymine-thymine dimer. Now, once those strong covalent bonds are broken, the only thing holding that damaged stretch of DNA to the other strand are the weak hydrogen bonds between the base pairing. So this is where that 4th protein comes along, UVRD. UVRD is a helicase. So it has the ability to break down the double-stranded helix. It breaks down those those hydrogen bonds between A's and T's and C's and G's. And so that removes the damaged strand. So now DNA polymerase 1, which is not the normal DNA polymerase. This is not the polymerase that does the vast majority of DNA replication in E. coli. But a DNA polymerase in either case comes in, uses the 3' end here as a primer, resynthesizes this DNA using the undamaged strand shown on the bottom as a template, and then DNA ligase creates that final phosphodiester bond, and you have what is hopefully a completely repaired piece of DNA, so that the lesion has been completely removed and the DNA has been resynthesized. So that's how excision repair works. Now, and as I mentioned, excision repair, and we're going to come back to a little bit about excision repair later. Normally, excision repair is something that occurs after pretty strong DNA damage. Thymine dimers are generally things that are made by strong ultraviolet light or strong chemicals. There are other mutations that occur during replication that need to be dealt with all of the time. So just a general background mutation occurs during DNA replication. When bacterial cells or your cells or my cells replicate DNA, even though we have proofreading activity, which takes care of most misincorporations, every once in a while something slips through the cracks. So this methyl-directed mismatch repair system is a system that's always present, and it's really based, or what it's trying to do is to find accidental misincorporations of DNA polymerase that have been skipped by the proofreading system. So E. coli as our standard example, but basically any bacteria does this, but we know the specifics of how E. coli does it. E. coli adds methyl groups to a particular 4-base pair sequence anytime it is seen. That sequence is GATC, shown here. There is a standard methylase, an enzyme produced by E. coli called, nicely enough, the DAM methylase. That DAM methylase is always doing this. That enzyme, anytime it sees the palindromic sequence, GATC, it's going to put a methyl group on the adenine of that. Basically, it marks it as self. In the same way that you and I mark our cells with major histocompatibility antigens so that our immune system recognizes self from foreign antigens, bacteria have really evolved pretty much the same type of a thing. They can mark their own DNA sequences with methyl groups so that they recognize foreign DNA when it comes in. Because bacteria are constantly worried about foreign DNA in the form of viruses, in the form of plasmids, or other parasites. Those are the two predominant parasites. So DAM methylase sees this GATC sequence, methylates that second A. Now, notice I said it's a palindrome. This is not a palindrome like the word radar, right? Don't think of it in that way. Radar is spelled the same forward and backwards. That's what you and I think of as a palindrome. GATC is a palindrome because this is 5'G, 3'C. Think about the other strand, the one that's not being shown, the one that's understood to be there. 5' to 3' on the other side, GATC. It's the same sequence. The beauty of this is that now that same 4-base pair sequence is going to be methylated at this adenine on both strands. So both strands are marked as self, eventually, not immediately. Because as I mentioned, this system that we're talking about now, the methyl-directed mismatch repair system, is designed to pick up misincorporations that have been missed by the proofreading activity of DNA polymerase. So that's what's shown here. So in this example, shown here in blue and gold at the top, you have a G and a T. Now you remember your basic base pairing rules. That's not a good match. If you are a DNA repair enzyme and you're shown this sequence, G and a T, without any other information, you have no idea whether to change this T into an A, meaning that the G was the correct mutation, or do you change the G into an A because the T was the correct one. You have a 50-50 shot of choosing that correctly. What this system does is it takes advantage of the fact that there is a window of time after replication whereby the DNA exists in a hemimethylated condition. The original strand that was that was used as a template is the older piece of DNA, and so theoretically is the correct form. And it's recognized as being the older form because this GATC sequence, as close as possible to this misincorporation, has got a methyl group on one strand but not the other. The DNA is hemimethylated. What happens is that the MUT SHL system, these are the proteins that make up this mismatch repair mechanism, first recognize the misincorporation. That's the role of the protein MUT-S. MUT-S senses that misbase pairing, that bump in the DNA, and will now serve as a nucleation point, a scaffold, if you will, to collect these other two proteins, MUT-L and MUT-H. Now that complex of proteins can now search out for the closest, most adjacent GATC sequence and then ascertain if one strand is methylated and the other one is not. It assumes that the strand that has not yet been methylated is the new strand, and so that one is the strand that logically contains an error. So it's probably the T, which was inappropriate here. So this MUT, H, S, and L will now remove that DNA between the closest GATC site and that misincorporation. After it's done that, DNA polymerase will simply use the other strand, the original strand, to resynthesize that entire stretch. The chances of it making the same mistake twice like that are infinitesimally small. So almost always this repair system is going to get it correct the second time. It's going to put in a cytosine opposite that guanine. So that mutation has been repaired. This system is actually important to understand because even though we're talking about it now in bacteria, it has a very strong homologue in humans. As a matter of fact, you can take the human MUT-S homologue and clone it into an E. coli strain where you have eliminated the gene for MUT-S, and it will actually replace MUT-S. And if you put in a defective human form of MUT-S, that E. coli will now have a much higher mutation rate. As a matter of fact, it's a natural mutation in that MUT-S homologue in humans that leads to as much as about 7% of human colon cancers every year. So this Lynch syndrome, or hereditary non-polyposis colon cancer, is a mutation in the human gene, which is analogous to the same gene in E. coli. So the same mechanism is occurring in our cells. And when our ability to repair those mutations is compromised, it can result in cancer. So it's important to understand how this system works because you're not simply understanding how bacteria repair DNA. Okay, so those are a couple of individual, I don't want to spend a lot of time going through individual repair mechanisms. What I'm much more interested in is how the DNA repair systems are controlled by bacteria, because there's a really fascinating global means by which bacteria control their repair system. And it's called the SOS repair system. SOS is not an acronym for anything, it just means save our ship. This is a response system that E. coli and other bacteria have evolved to catastrophic damage. So when things are really bad and the bacteria is up against the wall, it elicits this SOS response as a last-ditch effort to try and save itself. The way this system works is that in the face of a large amount of damage, it elaborates a large number of DNA repair systems, one of which is that UVR ABCD system, that excision repair system that we talked about just a few minutes ago. That system is normally expressed at very low levels in healthy cells. But one of the things that will happen when that cell gets dosed with a large amount of radiation or a DNA damaging chemical is the SOS response is going to turn on the transcription of UVR A, B, C, and D with the hopes that that repair system can fix whatever damage has been done. So that system, the UVR ABCD system, is actually controlled by this SOS response system. The response system, this SOS response, is basically all the result of what's known as the LEX-A regulon. LEX-A is a protein that has the ability to regulate a whole suite of genes, 30 or 40 different genes scattered throughout the bacterial chromosome. That's what's shown here in the top. So under normal circumstances, the cell is fat and happy. There's no no DNA damage to speak of. The condition would be as shown here in 1. The LEX-A repressor, shown by these red balls, binds to operator sequences to repress most of the genes that are involved in DNA repair, such as UMUDC, which we'll talk about. Here's UBRA. This is part of that excision repair system. So for the most part, when LEX-A is bound to these operators associated with these promoters, transcription levels of these genes are very low. They're not needed. The bacterium doesn't have a lot of DNA damage. So why bother to create these proteins when they're not needed? Especially since, as we're going to see in a little while, some of these genes, like UMUDC, while they're a repair mechanism, they're a hell of a repair system. They actually make mutations in order to create repairs. So you don't want this gene just going off at any time. So the LEX-A does a very good thing by holding these genes in check under normal, healthy conditions. But now, DNA damage occurs. So when DNA damage occurs, either by ultraviolet light or antibiotics that cause DNA damage, one of the ways that the bacteria senses that is that replication forks, the things that are trying to divide DNA, stall. So all of the enzymes necessary to replicate DNA can't get past DNA lesions, like thymine dimers. So they stall. And so what happens in a situation like that is there is a situation where single-stranded DNA persists inside a cell. Single-stranded DNA is not a normal situation for a bacterium. If single-stranded DNA persists to any level for any length of time, that's a sign that that cell has got DNA damage. Its replication forks are stuck, and it really needs to bring out the SOS response. It needs to break down that repression system, which is holding back all of those repair systems. So single-stranded DNA is really the alarm. What happens is that single-stranded DNA now has the ability to be bound by another protein, my favorite protein in all of bacterial physiology. You'll find out, we'll come back to this many times. REC-A. REC-A is a gene that is normally repressed quite well, but not completely, quite well by the LEX-A protein. So you can see it here being repressed. But some REC-A, a small amount, is present as a distant early warning system, if you will. Because now REC-A has the ability to sense, to find that single-stranded DNA. And when it does, it's kind of like Superman. It changes from this mild-mannered little protein that can't do much of anything. When it binds to single-stranded DNA, suddenly it develops a proteolytic activity that it never had before. It's got a big red S on its chest, and it's flying around. And now it has the ability, this protease activity, has an activity that is specific for the protein LEX-A. So now, when REC-A senses single-stranded DNA, a surrogate for DNA damage, it develops coprotease activity and begins to chew up LEX-A. Now, LEX-A begins to, as its levels decrease, the repression on these genes of the DNA repair system begin to fall apart. And so now genes like the UBR-ABC system begin to be expressed. As a matter of fact, the UBR-ABC system probably is the first system to be turned on. The SOS system is not an on or off system. It's not something that REC-A just chews up all of LEX-A and all of those repair systems are turned on at one time. LEX-A bound to these various operators has different affinities depending on the promoters. The LEX-A binds to the operator for UMUDC, for instance, with a very good affinity. Once that protein is on there, it doesn't come off very often. So it's not available to be hydrolyzed by REC-A very easily. As opposed to the binding site for Lexa in front of UVR ABCD. Here, that protein doesn't bind quite as well. So it comes off, it goes on. It comes off, it goes on. So it's available to be degraded quite readily by this REC-A coprotease. So the first genes that are going to be turned on, that are going to become de-repressed, are going to be the excision repair mechanism. The bacterium hopes that that system by itself is enough to repair the DNA. And if it does, single-stranded DNA goes away, REC-A becomes calmed down, it stops chewing up Lex-A, and the SOS system resets itself without ever having to bring out the big guns, the UMUDC. That's what we're going to talk about on Wednesday when we come back, the damage that these repair mechanisms can actually occur after the SOS control system has lost. I'll see you guys in the lab, and I'll see you on Wednesday.